Hello, in this video we're going to take a look at the embryonic development of the central nervous system, and the way we're going to do this is we're going to use some 3D models which are absolutely brilliant and have been developed by Dr. Bernadette de Bakker over in the Netherlands. These are based on real embryos, and they're based upon serial sections taken of real human embryos. So they're entirely accurate and not idealized in any way. So we're going to go straight through a number of different stages of development and try and correlate that with some disease states that might occur as a result of disordered development. Just for your reference, when we're looking at the screen, there is a standardized staging system called the Carnegie Stage. However, I prefer to think in terms of the number of days old that the embryo is. It's given as a range. And even more interestingly, we're also told the size of the specimen. So in this case, this embryo here is only 0.6 millimeters long. So just keep your eye on those numbers and you'll be able to get an appreciation then of how small some of these things are that we're talking about. So let's start off and let me just explain what the view is we've got here. Here we're looking down onto the primitive disc, the embryonic disc. And just to orientate you, the top here will become the head, and the lower portions will become more caudal regions of the body. And we're looking down at the superior side of this embryo, and these blue cells we're looking at here are the epiblast cells, which will become a number of different cells, but these ones we're looking at are going to become the ectoderm, which ultimately will become the skin and central nervous system of the adult body. Now, when we're looking down onto the embryonic disc, what we start to see at around about this stage, at about 18 days of age, is that we develop a slight indentation here called the primitive streak. And at the top end of the primitive streak, we have a little pit called the primitive pit. And what's happening along this primitive streak here is that we have a process of invagination going on. And this is causing the process of gastrulation to occur. And what's happening is that the epiblast cells here are approaching the primitive streak and they are invaginating. And in doing so, they are going on to form the other two embryonic layers. So if I just change the angle here a little bit of this, what is happening is that these epiblast cells, we'll call them ectoderm cells from now on, are approaching the primitive streak, they're invaginating, and then they're migrating in all directions to form the two additional embryonic layers. We have the mesoderm in red, and we have the endoderm in green. And all three of these embryonic layers are derived from this epiblast, okay, which itself goes on to form the ectoderm of the embryo. So we formed the trilaminar embryo, and this is very important because from each of these three layers, all of the body tissues will go on to develop. Now, a very important part of the mesoderm grows cranially from the primitive pit here, and that is the notochord. And if I just hide the ectoderm, we should get a chance to see the notochord. Okay? So here we can see in pale blue the notochord, which grows cranially from the top of the primitive streak. This notochord is really, really important because it is the notochord which is going to go on to induct the overlying ectoderm and essentially tell it to start forming the neural tube and hence the rest of the central nervous system. In the adult, very little is left of the notochord. All that's left in the adult is the nucleus pulposus in the intervertebral discs. This notochord is really, really important for inducting the overlying ectoderm to form the neural tube. So let's move on now a little bit. Let's move on to the next stage in the development, where we're looking at an embryo of approximately 20 days old, and now it's about 1.5 millimeters long. What we're looking at here is it's entirely ectoderm that we're looking at. All right. So in this pinkish colour here, we can see the ectoderm that's going to go on to form the skin. Whereas in green, we're looking at the neuroectoderm, the ectoderm that's going to go on to form the neural tube. And what you can start to see is happening is that the ectoderm is starting to elevate at these regions here. The ectoderm is starting to elevate at this region here and this region here. We can get a better appreciation of that maybe if we look at a cross-section. So, for example, if we take a look at a cross-section here, we can get a bit of an appreciation for how the ectoderm is rising up. These are the neural folds, okay? So where the ectoderm is folded to form these two ridges, we have the neural folds, and in between them we have the neural groove running longitudinally. So, and what we're seeing here is a process of invagination. We are seeing, under the direction of the notochord, the ectoderm starting to rise up, fold, and then invaginate down. And it's going to start to form a tube. It's going to start to form the neural tube. Now, if you look at this neuroectoderm, you notice at the cranial end, it's much more elaborated than at the caudal end. And that perfectly correlates with the fact that this end is going to form the brain. So it's going to be bigger and more sophisticated than the more caudal end. The epidermal ectoderm here has been rendered partially transparent, and that affords us a view of the notochord running longitudinally. So this notochord has been releasing a whole load of chemical factors which have been inducting that overlying ectoderm to start to fold and buckle in order to form that neural tube by invagination. Now, in this next stage, it's around about 22 days of age and 2 millimeters long. We see something interesting has occurred. Once again, we've got the ectoderm here, which is going to form the skin in pink, and we've got the neuroectoderm here in green. And notice now what has happened. What has happened here is that the neural tube at this point has fully invaginated. The neural tube has fully invaginated. And if we look at a cross-section at that level, we can see that we have now very, very nicely formed a complete tube, and that the ectoderm, the ectoderm that will become the skin has healed up over this point and reconstituted the body wall. So at this point, we have formed a complete neural tube. Now, this is the stage at which things can go wrong. What you've got to imagine is that this neural tube, starting from this region, which in the adult corresponds to the cervical region, this neural tube is going to zip up along its length. It's going to zip up cranially to form the brain, and it's going to zip up caudally to form the spinal cord. If the neural tube fails to close in the cranial direction, this leads to the extremely severe condition known as anencephaly. Because if the cranial neural tube does not go on to close, then it won't form the brain. And this is usually incompatible with life. Okay? These children don't normally live for very long at all after birth because their brain simply has not formed. If the neural tube fails to close in the caudal direction, this leads to the condition known as spina bifida. Okay? And spina bifida has a number of different forms, which we'll talk about further in other videos. But spina bifida essentially is failure of the closure of the neural tube in the caudal direction. One final thing that we should consider before moving on, if we once again look at a cross-section here, where these neural folds come together, essentially at the apices of the neural folds, we have a further population of very, very important cells in development. And these are known as the neural crest cells. And what you've got to imagine is once the neural tube has closed, these neural crest cells become free, and they then migrate to a whole load of different places within the embryo to contribute to a very diverse set of tissues. For example, they contribute to structures in the head and neck, to structures in the heart. They form the melanocytes within the skin, and they form a whole load of sensory and autonomic ganglia as well. So the neural crest cells that develop from the apices of the neural folds are very important and contribute to many, many tissues within the embryo. So now let's move on to the next stage. And in this stage, what we're looking at here is that there has now been complete invagination of the neural tube in both the cranial and the caudal directions. We're at about 26 days post-conception. And this is still only about 2.5 millimeters long. So this is very, very small. Very, very small indeed. Excuse me, I just accidentally pressed the wrong button there. So, okay, so I'll just restore the view. There we are. So it's still very, very small, but the crucial thing that's happened now is that we have closed the neural tube entirely. Also on this view, we can see some of the somites here. These are made from mesoderm and go on to form components of the skin, the vertebrae, and also muscles as well. What I'm going to do, though, from this view is I'm going to extract just the neural tube. So by clicking on this, we can extract just the neural tube and isolate it from the other structures. So this is the neural tube, and you can see at this stage the neural tube is still really quite simple. It's not particularly complicated and bears very little resemblance to the adult central nervous system. However, if I zoom in on the head end of this, we can start to appreciate that something interesting is happening. The first thing I want you to appreciate is that the cranial end of the neural tube has developed 3 swellings. So there's one swelling here, there's one swelling here, and there's one swelling here. These 3 swellings correspond to the 3 major divisions of the brain. This swelling will become the forebrain, this swelling will become the midbrain, and this swelling will become the hindbrain. Okay, so at this stage, at around about 25 days, we can see that there are the 3 brain vesicles. If we look at the forebrain vesicle, we can appreciate something more. We can see that it is actually starting to go through a process of producing laterally directed outgrowths. And these outgrowths depicted in grey are in fact going to become the optic nerves. Okay, so we can start to see that formation of the optic nerves, and these are growing towards the developing eyes to connect up with them. So we're starting to see the 3 divisions, and additionally we're starting to see some flexions appearing. We can see that there is a flexure here at this point, just above the midbrain. This is called the midbrain flexure or the cephalic flexure. Furthermore, we can see a flexure has started to very subtly appear here, and this is the cervical flexure at the junction between the brainstem and the presumptive cervical spinal cord. So we can see brain vesicles, and we can see flexures starting to develop. Now we've moved on. How far have we moved on? We've actually moved on quite a long way, about 10 days now. And essentially what's happened is we've had further elaboration and growth of the various organs of the body. However, as far as the central nervous system goes, in that intervening 10 days, no seriously major events took place. That's why I haven't shown you any other intervening stages. And really, it's quite beautiful what we're looking at here. We can see in green the neural tube. We can see the vascular system starting to form in purple and in red here. We can see the heart starting to develop and the liver. And furthermore, we can see cranial nerves here in orange and spinal nerves here in orange down at the caudal levels. So there is a lot going on within this embryo. But because this is focusing in on the nervous system, let's focus down onto the brain vesicles once more. But in fact, what I'm going to do is not just show you the brain vesicles, I'm going to show you all of the nervous system structures. So what we'll do is we'll blank everything and then we'll bring in all of the nervous system structures. So we can see the neural tube, we can see the brain vesicles and the flexures and the spinal cord down here. But I want to just zoom in on some structures here. We can start to see now, corresponding to each somite, a dorsal root ganglion and a spinal nerve has started to develop. Furthermore, in blue, we can see the sympathetic chains have very nicely formed. These sympathetic chains, along with the dorsal root ganglia, developed from the neural crest, which originated back here and then migrated ventrally, forming the dorsal root ganglia and then the sympathetic ganglia themselves. So we've got the sympathetic chains here, the dorsal root ganglia and the spinal nerves. Moving up, zooming in a bit, we can see the tremendously complex cranial nerves now forming and innervating the various pharyngeal arches. For example, very nicely we can see our trigeminal nerve just there and all the other cranial nerves. We can see also the vagus nerves here descending down into the thorax. Let's remove these distracting structures and let's now focus down on the brain vesicles once more. And the reason that we've got some structures in dark green and some in pale green is because the brain vesicles now are separated into some further subdivisions, which we're going to look at. But let's just look at the simple things first. We can see that we have the forebrain vesicle, the midbrain vesicle, and the hindbrain vesicle. Furthermore, we have the spinal cord here. We can see that the forebrain has in fact started to form two lateral outpouchings. These are different now to what forms the optic nerves. These are the outpouchings which are going to form the cerebral hemispheres. Now, the different vesicles also have flexures separating them. Here is the cephalic or midbrain flexure, and here is the cervical flexure corresponding roughly to the hindbrain spinal cord boundary. The parts of the brain vesicles have actually been separated. There are subdivisions within them. The forebrain, the true term for the forebrain is the prosencephalon. The prosencephalon. And the prosencephalon is differentiated into two subdivisions. Most cranially, we have the telencephalon, which is going to form the cerebral hemispheres. And sitting just behind it is the diencephalon, which is going to form the thalamus, the hypothalamus, and the optic nerves. So the prosencephalon has the telencephalon and the diencephalon just behind it. Then with the prominent flexure, we have the midbrain or the mesencephalon. So meso means middle, corresponding to the midbrain. The midbrain only has one division. And then the hindbrain is strictly more accurately termed the rhombencephalon. The rhombencephalon refers to the hindbrain. And the hindbrain or the rhombencephalon has two subdivisions. The metencephalon, this pale region here, goes on to form the pons, and you can see the bulge of the pons just about there. So the metencephalon forms the pons and the cerebellum, whereas the myelencephalon goes on to form the medulla. So that is quite confusing. Those terms can be quite confusing, but essentially, if you just stick to forebrain, midbrain, hindbrain, remembering that these vesicles are going to form the cerebral hemispheres, this is the midbrain, and this is going to form the thalamus. You shouldn't go too far wrong. So that's very, very nice demonstration of the vesicles, how they're developing and their corresponding flexures. Now let's move on again. So we're at about 35, 36 days here. We're going to move on now, once again, quite a bit, to about 45 days. And here we are looking once again at primarily the nervous system structures. And really, a tremendous amount hasn't occurred, different to what we saw before. But the key message I want to give you is that we have had further elaboration of the neural tube, particularly up at the cranial end. Now, very nicely, you can see that the telencephalic outgrowths are forming structures that look very reminiscent of the cerebral hemispheres. We can appreciate that here we've got the diencephalon, and if I zoom in, you can actually see the optic nerve there growing out from the diencephalon to the eyeball. We can see the cephalic or midbrain flexure here. We can see the very prominent metencephalon, part of the hindbrain, with its trigeminal nerve and its three major branches there. So the three branches of the trigeminal nerve are very clear there. We can even see the inner ear structures just there. All right. So once again, this is just a revision of those brain vesicles. Another thing I want to emphasize to you is I want to add on the skeletal system. And the skeletal system is not fully developed yet. But what I hope you can appreciate just by looking at the derivatives of these somites is that still at this stage, we're about a centimetre long, the neural tube fills the entirety of the vertebral canal. So at this stage, there is pretty much a one-to-one correspondence between spinal cord segments and vertebral levels. But what's going to happen, as you know, as growth proceeds, the vertebral bodies are going to grow much faster than the spinal cord, and this will result in the chorda equina being pulled down considerably, leading to the formation of that big bundle of nerve filaments. Now, finally, let's take a look at an embryo which is at around about 2 months of age. It's about 3 centimetres long, and we can really start to see some remarkable things are happening. We can see that the brain is really very nicely developed here. We can start to see a cerebellum starting to grow. We can see cerebral hemispheres very clearly, the spinal cord with the dorsal root ganglia, and we can even see that the peripheral nerves are growing into the upper and the lower limbs. If I add on the skeletal system, you can see very clearly, as I said earlier, that there is a one-to-one correspondence pretty much between vertebrae and the neural tube, but that as the vertebrae, particularly at the lumbar levels, grow, then they will pull down the dorsal and ventral roots, forming the chorda equina. One final thing I want to do before we wrap up is I just want to show you what the ventricular system looks like in a 2-month-old embryo. And now we can zoom out a little and we can look at the ventricular system. Remember that the ventricular system is corresponding to the lumen of the neural tube, and developmental defects in the ventricular system can cause things like congenital hydrocephalus. So if there is blockage of the drainage of CSF, we can develop hydrocephalus. We can see corresponding to the cerebral hemispheres, and really quite large, because we haven't had appreciable growth of the neural tissue. So these ventricles are filling up a large cavity within the brain. These are the lateral ventricles. Then we can see the third ventricle just around about here in the midline. Then we can see the cerebral aqueduct here, once again Everything seems bigger in the ventricular system than in the adult, just because the neurons haven't had chance to proliferate. But this is the aqueduct. And then back here is the fourth ventricle going down to the central canal of the spinal cord. And we can start to appreciate that we're getting a foramen of Magendie developing there, and our foramina of Luschka sitting more laterally. So that's all I wanted to say about the development of the nervous system using these 3D videos. Thank you for listening.